Hi, this is Magdi Isa. I'm your teacher for Anatomy and Physiology Bio 201 Lab. This video, just like all of my other videos, is intended to be only for Grand Canyon University students who are enrolled in one of my classes. Please do not share this video with any other students who is not enrolled in any of my classes or not a part of Grand Canyon University institution. So today we're going to discuss different types of tissues, and as we know, tissues are formed of cells. Tissues come together to form an organ. So the four main types of these tissues are epithelial tissue, connective tissue, muscle tissue, and nervous tissue. We will discuss today only epithelial tissue. Next time we will discuss the connective tissue. When it comes to the muscle tissue and nervous tissue, these ones will be discussed in the second or third units. So the main function of the epithelial tissue is actually covering and lining of different structures. Connective tissue is responsible for support, while muscle tissue, as we know, is responsible for movement. Nervous tissue is responsible for control. If you need a little bit more details, then the epithelium lines, protects, and secretes. The connective tissue is living cells in non-living matrix. Muscle tissues are excitable, contractile tissues for movements, and nervous tissues are excitable tissues used to send short-term signals throughout the body. So let's start with the epithelium. So the epithelium covers the body surfaces and lines the body cavities, and it makes most of the glands. The main function of the epithelia is to do protection, is to help with the diffusion, absorption, secretion of ions. It's also responsible for filtration, and it has some other functions. I'm not concerned too much about you knowing the function of epithelia. I just need to discuss the basic types and the special criteria of epithelia today. So special characteristics of epithelium. The epithelium, or epithelia as plural, has a lot of cellularity. What does this mean? This means that cells are separated by minimal extracellular material. Extra means outside the cell. So cells are closely packed together, and this is really different from other tissues like the connective tissue, where you have cells are completely separated from each other and there's a big space between the cells. So this is number one, is cellularity. Number two is specialized contacts. Since these cells are close to each other, they will be connected to each other by special junctions. We will talk about these junctions in a second. Number three, which is very important, is polarity. Polarity means that not every side of the cell is the same. Not every side of the cell is the same, meaning that you have an apical surface, you have a basal surface, and you have also a lateral surface. All epithelia are supported by connective tissue, and all epithelia are avascular. A here means no, so the letter A or N in front of a word without a space means no. It's not vascular, but it has innervation. So it has blood. It has no blood vessels traveling through it. It gets its blood supply from the connective tissue that's below it, and it gets its innervation travels also through the connective tissue and through the epithelium. And the last point is regeneration. As we all know, if you scratch your skin, you get this epithelium which covers your skin. You get it back quickly because the epithelium has a very high ability to regenerate. And if you compare this to for example, the nervous tissue, if you cut a nerve, this nerve has a very small chance of being repaired. Okay, so here is just one example of an epithelial tissue. So this is one cell, this is the second cell, this is the third cell. These cells, they are supported by connective tissue. So this is my connective tissue, this is my epithelium. As we can see, this is the apical surface of the epithelium. This is the basal surface of the epithelium. And these two are the lateral surfaces. And as we know, these are different from each other. You can see on the apical surface there are some elevations. We call these microvilli. And the base here is sitting on the connective tissue by something called the basement membrane, which we will talk about in a second. Just above the apical surface, we have the lumen. The lumen is an empty space that does not have any cells in it. So what else we have? As you can see, there are different types of junctions between cells on the lateral sides. And as you can see also, the blood vessels is traveling in the connective tissue, but it does not travel through the epithelium. The epithelium gets its nutrients by diffusion, simple diffusion, which we'll talk about in Lab 4. And the innervation, as you can see, the nerves actually travels through the epithelium, so the epithelium is sensitive. So what I want you to think about the epithelium just like a mountain. So you have an apical surface and you have a basal surface. And if we just flip the picture, well, here is still my apical surface and here is still my basal surface. And this is my lumen. Why am I saying this? Well, because if you look at one slide from the kidney where you have epithelium, so this is my epithelial tissue. This is a cell here. I know it's a cell because I can see its nucleus. So this is my basal surface, this is my apical surface, and this is my lumen. On the other side, this is still my apical surface, and this is my basal surface, even though it's upside downward, because the apical surface is the surface that is closer to the lumen. And all the apical surfaces are going to have the same specialized characteristics, like the microvilli, for example, and the all basal surfaces where they will have also the basement membrane, which is specific for the basal surface. So let's talk about polarity. We said we have basal, apical, and lateral surfaces. So let's start with the basal surface. On the basal surface, we have the basement membrane, which is typically formed of basal lamina and reticular fibers. The basal lamina is secreted by the epithelium, while the reticular fibers are secreted by connective tissue. So the basement membrane is actually made of both of them. Some people do not make a big distinction between basal lamina and basement membrane, and I would not also make a big deal out of that. So just know that the basement membrane or basal lamina is related to the basal surface of the epithelium. When it comes to the apical surface, we have two important structures. We have a microvilli and we have cilia. Microvilli are the finger-like extensions from the plasma membrane, while the cilia are the wipe-like structures. They are very highly motile. So what is the function of the microvilli and where do we find them? Well, let's suppose that we don't have any microvilli here. So let's suppose that the cell just ends here. And now let's compare this to another cell that has microvilli. Well, all these microvilli increase the surface area. And why do we need to increase the surface area? Well, in some areas like the GI or the kidney, we need to increase the absorption. So if we increase the surface area, we'll have more contact with the contents in the lumen, and this increase in surface area will make the cell more efficient in absorption. How about the cilia? How they're different? Well, these ones actually are very motile, and they push things. Where do we find them? We find the cilia in our respiratory system. So this is a layer of mucus. This is our cilia here on top of the epithelium. So these ones, they move the mucus towards outside. So if you have any bacteria comes here and gets stuck to this mucus, or any viruses or any dust, our cilia will push them out, so you can either spit it or swallow it. Next is the lateral surface. We have three different types of junctions. We have tight junction, desmosomes, and gap junctions. The tight junctions and the desmosomes, both of them, are responsible for attaching the cells together. But the gap junction is actually kind of a connection between the cells that allows ions and small structures to travel between the cells without having to exit the cell and enter the other cell. So again, tight junctions, desmosomes, are for connecting the cells. Gap junctions is for having the cells communicate with each other, so the ions can travel between them. And this is as far as I want you to know. Now, the classification of epithelium. The epithelium, just like you and I, they have first name and they have last name. The first name is either simple or stratified, and the last name is either squamous, cuboidal, or columnar. So let's go to this slide here. So these all are simple epithelia. What does simple mean? Simple means one layer. These all are stratified epithelia, meaning that it has more than one strata or more than one layer. So this is the first name for the epithelium. What's the second name come from? The second name comes from the shape of the most superficial layer. So I have simple, squamous. Squamous means flat. Simple, cuboidal means like a cube. And columnar means that it's like a column or it's rectangular in shape. How about this one? This one is stratified. Well, what's the other name? Do I call it cuboidal or squamous? Well, again, I said the second name comes from the shape of the most superficial layer. So it's stratified squamous epithelium. This one is stratified cuboidal because the second or the top layer is cuboidal. And finally, this one is columnar because the most superficial layer is columnar, even though we have cuboidal cells here, but they are at the base. So we have simple epithelium, stratified epithelium. The simple is simple squamous epithelium, simple cuboidal, and simple columnar. And there's something also called pseudo, which means false. Pseudo stratified, which means multiple layers. So it's false multiple layers. What does this mean? This means that it's one layer because it's false multiple. It looks like it has multiple layers, but it's not. And the stratified, we have stratified squamous, stratified cuboidal, and stratified columnar, and we have the fourth type is transitional. So let's start talking about the simple epithelia first. the simple squamous epithelium. So it's just one single layer that is flat, and the nuclei are also flat. Why do we need one layer that's flat and it's very thin layer? Well, because we need this in areas where we need material to travel. So it's much easier if you have one layer that is really very skinny. Where do we need this? We need this in the renal corpuscles, which is a place in the kidney where the filtration process happens. Basically, the fluid travels from the blood to the lumen of the kidney tubules, so it can become urine later on. So we need a lot of area that has a small height. It's also in the alveoli of the lung because this is the place where gas exchange to happen. You want only very thin layer. And it lines blood vessels and lymphatics. Guess what? You actually have the diffusion of nutrients from the blood to the cells occurring through the wall of the blood vessels. So you need this layer to be very thin as well. So here is the picture that I want you to know, and I want you to know actually all the pictures on this lab. I have here, this is one of my air spaces called alveoli in the lungs. And these alveoli, as you can see, they are made of very thin layers of epithelium, and every one of these violet or these violet dots is actually nucleus. So this is a nucleus, that's a nucleus, that's a nucleus. Okay, so these are all three different cells in this area. And as you can see, they're very thin, so the gas exchange can occur very easily between blood vessels, that looks like a blood vessel here, and the alveoli. The second type is simple cuboidal epithelium, and this is just looks like a cube. The nuclei will be rounded. Where do we need this? Well, we need this in a place where the cell has to be a little bit bigger, so we can do either secretion or absorption. An example of that is kidney tubules. So here is the kidney tubules, and we have, as you can see, cells here that are cuboidal in shape, and here are the nuclei that are rounded. And of course you cannot see the nuclei of these cells because this is just two planes, that's a cross section. So the nucleus could be either higher or lower than the level of this section, so that's why you don't see it. But every cell, of course, would have to have a nucleus. Now the blood vessels are here. So what happens through this one is we need one single layer to allow for the transport, but at the same time there is a process that occur inside these cells, so we need to have these cells a little bit taller. Next type is simple columnar epithelium, and this is the only type that, not the columnar cells are the only ones that have cilia and may have goblet cells between them. The same thing, we also need just one layer to allow for absorption, but at the same time there is a process of secretion and transport and some changes that occur on the molecules inside the cell, so we want the cell to be somewhat taller. So there are two types of the simple columnar epithelium. We have non-ciliated forms that do not have cilia, and this is in the digestive tract, and we have the ciliated forms, which is in the bronchi because we need to expel the mucus, and in the uterine tubes because we need actually to move the ovum. Okay, so we need to have these cilia so we can move the ovum. So here is an example from the GI tract. As you can see, these cells are very tall, and these cells have nuclei in them. These cells are not ciliated, they just have some microvilli, but they're not ciliated at all. Last type of the simple epithelium is called pseudostratified columnar epithelium. And why it's called pseudostratified? Well, first, it's located into two places. The ciliated type is in our trachea, and the non-ciliated type is in the male reproductive system. So let's see this one. Why they called pseudostratified? Well, because under the microscope you cannot see the lines between the cells. So you actually look only at the nuclei to determine if there is a cell at this point or not. So when you look at only at the nuclei, you can see this might be a layer of cells, this might be a second layer of cells, this might be a third layer of cells, this might be a fourth layer of cells. But when you look at the electron microscope, you can see actually that all these cells actually start at the basement membrane. Some of them reach the top, like this one, and some are very short, do not reach the top. So where do we have the nucleus? We have the nucleus in the widest part of the cell. So this cell, this is the nucleus of that cell because this is the widest part. This is the widest part of the cell. So why these cells are not stratified? Because it's basically one layer. All of them start at one level. Some are tall, they will reach the top. Some are short, they will not reach the top. That's why it's just one layer. But pseudo, because it looks like it's stratified, but of course it's not stratified. I hope this helps. If you have any questions, please let me know in the classroom. The next type is actually going to be our stratified epithelium. The stratified epithelium, either stratified squamous epithelium. And why do we need this type? Well, we need many layers. And why do we need many layers? We need many layers in places that require protection. So we have two types. We have a type that is keratinized, and we have a non-keratinized type. So what is keratin? Keratin is basically a layer of secretions that is made by skin cells. So where do we find this keratinized stratified squamous epithelium? This is present in the skin, epidermis of the skin. The non-keratinized type is present in lining of the mucous membranes and body openings. So in areas where there's a lot of friction, example of that is your mouth where you're eating all the time, anus because you're pooping every day, esophagus because also food travels through the esophagus, vagina, there is friction, urethra is the same thing. So here is an example of these cells. As you can see, the nuclei here are rounded because these cells at the base, are more or less cuboidal or rounded cells. But as you go to the top, look what happens to the nuclei. They become flatter, and then they become very flat, slit-like nuclei at the top. And is this keratinized or non-keratinized? I do not see keratin here, so this is non-keratinized stratified squamous epithelium that comes from the esophagus. The stratified cuboidal epithelium also has protective function because it's two or more layers of cuboidal cells, and it's present mainly in large ducts of mammary and salivary glands. As you can see here, I can see only the epithelium, So I can see only the nuclei. So here is one layer of nuclei that are rounded, which means that this is one strata or one layer of cells. Here is a second layer of nuclei. So this is a second layer of cells. So I have two cell layers here. Nuclei are rounded, this means that the cells are cuboidal. And as we said, we find these in our salivary glands. Stratified columnar epithelium. It's a rare type of epithelium and is present probably only in the male urethra. So let's see. Again, I have one layer, then I have a second layer, third layer, maybe fourth layer. But what's the highest layer? What's the most superficial layer? What's the top layer? It's columnar. So we call this stratified columnar epithelium. How do I know from this slide that it's columnar? Well, because the nuclei are oval shaped. It's not flat and it's not circular, because the flat one is the ones that are in the epithelial squamous epithelium. The circular ones are for cuboidal epithelium, and the oval ones are for the columnar epithelium. The most interesting type of epithelium is actually the transitional epithelium. It's also called uroepithelium. And why it's called uroepithelium? Because basically it's in your urinary system. It's in the urinary bladder, the ureters, and the proximal urethra. So let's take a look. So that's how it looks like. You have some cells that are maybe rounded down there, and then at the top you have cells that are big and they're rounded and weird shaped. So here is one cell, second cell, third cell, and fourth cell. Why this is called transitional epithelium? Because this epithelium is actually in transition. It changes its shape. So unlike all other kinds of epithelia, this one changes its shape based on the state of the urinary system. So let's see. So this epithelium will look like this in one time. It's very tall epithelium. It has 1, 2, 3, 4 layers. And it might become flatter at certain time. So 1, 2, 3, 4 layers, but they're all still shorter than this one. So if this is your urinary bladder, I want you to stop the video at this point and tell me, which state is this? Is this your urinary bladder when you are full or empty? And which state is that one? Is this urinary bladder when it's full or when it's empty? Okay, I hope you had a chance to stop the video and think about this. The answer to that is the following. This is actually your urinary bladder when it's empty. So if you have a small bladder like this, there is a high height for the epithelium, and this is your urinary bladder when it's full, because when you have a big bladder, it's full of urine, it's so stretched, so if it's stretched, you will have a smaller height for the epithelium. I hope this makes sense to you. So this is just a review for you. Every time, I hope you're gonna stop the video to ask yourself, what type of epithelium is that? So let's start with the first question. What is this type of epithelium? Well, this is simple squamous epithelium. How do I know this? Well, I can see the nuclei, and the nuclei are flat. So if nuclei are flat, this means that it's just squamous cell, and I see only one layer. That's why it's simple. Let's go to the next one. How about this one? Well, it's one layer again, and the cells are, the nuclei are rounded. This means that the cells are cuboidal. So this is simple cuboidal epithelium. How about this one? Again, the nuclei are oval, and you have a tall cell. So this is a simple columnar epithelium. How about this one? Well, this one's a little bit hard, but all cells start at the basement membrane. Some of them are very tall and they reach the top. So this is pseudo-stratified ciliated epithelium because I can see the cilia here. How about this one? Well, I can see nuclei that are rounded here. As I go to the top, nuclei gets flatter and flatter. So this is stratified squamous epithelium. It's actually also keratinized because here is a layer of keratin. And actually this is comes from the skin. So this is the epidermis. So again, the correct answer is keratinized stratified squamous epithelium. How about this one? Well, again, I see nuclei that are rounded. I go to the top, they become flat. I don't see keratin, so this is non-keratinized stratified squamous epithelium. Next one. Well, I see two layers. There's one layer here, there's a second layer here, and these cells have rounded nuclei, so these are stratified cuboidal epithelium. Next one. This is our transitional epithelium that is having dome-shaped cells on the top, and we have somewhat taller cells at the bottom. How about this one? So this one is also a difficult slide, but I see a layer here of cells, second layer, a third or fourth layer here. So these nuclei are oval, which means that these cells are going to be tall cells. They're columnar cells. It's multiple layers, so it is stratified columnar epithelium. The last part of this lecture is going to be the glands. And we have two types of the glands. We have endocrine glands and exocrine glands. What are the endocrine glands? Are glands without ducts. They're called ductless glands. The exocrine glands are glands that do have ducts. Endocrine glands secrete hormones. Exocrine glands, for the most part, they secrete enzymes. However, they can secrete other things other than enzymes, like, for example, sweat glands, they secrete sweat. Sebaceous glands, they secrete the sebum. Salivary glands, they secrete the saliva. Mammary glands, they secrete the milk. Liver secretes bile. And mucus-secreting glands, they secrete mucus, while the pancreas secretes the enzymes. Okay, so let's see some examples of the major endocrine glands. We have different types of glands. I just want you to be familiar with the names of these glands. At this point, you're not required to know the function of any of these glands. Now, we classify the exocrine glands into either unicellular glands. Uni means one, cellular means one cell. We have only one example, goblet cells. So goblet cells is just a cell that's by itself a gland, considered to be a gland. And we have multicellular glands. The multicellular glands are going to be classified further, which we'll do that in a second. But let's see first, what does the unicellular glands do, or what does the goblet cell do? It secretes mucin, which attracts water and makes mucus. So this picture here shows us goblet cells. Here is goblet cells under the microscope. These cells are full of mucus. The next type of cells are the multicellular glands, exocrine glands, and these ones are much more complicated. They have ducts and they have secretory units, and we can either classify them based on their ducts into simple or compound, and based on their secretory unit into either tubular, alveolar, or tubulo-alveolar. Let's see how does this look like. Well, here are different types of glands. Let's start with this one. First, is this one duct or is it multiple ducts? Well, it's only one duct, so it is simple. The same way here, it's also simple. These guys have multiple ducts. Here, all this yellow part are the ducts, and the pink part are the secretory units. So this is going to be compound. Now let's talk about the secretory unit. Is it going to be just a secretory unit that it looks like a tube, or it's more circular? So if it's a tube, it's going to be tubular. If it's circular, we will call it alveolar. So it's simple, tubular, because simple means one duct. Tubular means the secretory unit is tubular. Simple, alveolar, because simple means just one duct and alveolar means a rounded shaped secretory unit. Same thing here. Compound ducts. Well, compound means multiple ducts, and compound tubular means that secretory unit is tubular. This one here is going to be compound alveolar because all the secretory units are going to be circular, and tubulo-alveolar because some of these are like tubules, some of these are like rounded. I hope this is simple enough. But now let's add two more types to the picture. If we start with this one here, now look at the duct. Is it one duct or more than one? Well, it's just one, so we're still simple. But what's happening to the secretory unit? Well, it is actually more than one. It's branched. So it's simple, branched, tubular. The same for the alveoli. So it's simple, branched, alveolar. Because what's branching here is not the duct. It's actually the secretory unit. Okay? Now let's go to the next slide. Well, this slide comes straight from your textbook, and it has the same exact ones that I just spoke about, but there is an extra type here. It is, look at this one first. So ask yourself, what is this type? It's simple, tubular. How this is different from that one? Well, that tubular part is actually coiled, so we call it simple, coiled tubular. The next question here is, well, how do we actually classify the glands? Not by their structure, but by the way of secretion. And this is a very important concept that I want you to know very well. We have a merocrine secretion, merocrine. We have apocrine secretion, apocrine. And we have holocrine secretion, holocrine. How these are all different? Well, the cells start the same way, but the secretion is going to be different. Let's start with this one. What happens to this cell that is secreting by the holocrine method? Well, holo means all. So after the cells creates and synthesizes its own secretion, it's going to just explode, and everything will be just released into the duct. So that's it. The cell is gone. There's no more cell. The cell just exploded. An example for that is our spacious glands. How about the apocrine? Well, apo from apex. What happens is the cell creates the secretion, push it towards the apex, and the apex is going to be cut and released. And the cell, of course, will regenerate and become like it was before. So in this case, the cell does not die. It's just the apical part of the cell that dies. And this occurs in mammary glands. The last one here is going to be the merocrine secretion, and most of our glands secrete this way, which is basically you make secretions and you release these secretions by exocytosis. And one example only is the pancreas, and this is how the pancreas secretes its enzymes. Please make sure that you understand this concept, otherwise ask me all questions in the classroom. So I'm done with my part. You have now the quiz that you can take care of. Thank you for watching this video and have a great day.